There goes a little.
You got a jelly,
Yeah.
a midwater schmutz. Like how I've declassified everything you love into just schmutz. It's terrible.
Yeah,
I'm a terrible
all
lead.
those quadrillions of organisms out there.
Ooh, I see bottom.
Yes, we see bottom. That's exciting.
Yep, sand. Really wavy sand.
Oh, what is that?
I don't know what that white thing is. Looks like a rock. It's very disorienting.
Yeah, now it looks... Well,
The sand's probably pretty thick here. Oh.
And there's a
fish
in the bottom of the
Oh, there you
view
go.
here. Is that a shark?
I don't know.
I don't think so. Nice. So we are 3 meters from the bottom and spotted our first living creature. After 2 days of not seeing fish, I'm sure the the fishy people are very happy. Nutrient poor waters that we were visiting the last couple dives.
Now that our pilots get settled, and then we'll get the okay to start zooming.
Just a reminder that we'll be wanting another water sample after we get situated and get going.
You can see the waves in the sediment here. There's lots of current on bottom. I'd imagine a lot of the sand is just spilled down from the surface.
Right.
Because it looks like there's just this featureless area that we're in, except for this one sinkhole we're sitting in. So the sand probably just pours down the sides and into the bottom of this.
And these ripples aren't generally aligned.
Yeah, they're really mushy.
That it's, current is changing directions down here from time to time.
Oh my goodness, there's a soccer ball.
Well, we
I can't believe that.
we did hear that there was going to be a possibility of marine debris accumulating.
Soccer out here.
Well, it just shows how far our waste can go.
Yeah, it probably floated out here, yeah. And just eventually sunk. Jeez. Yeah, right? I think it would probably fill with water, and then that's why when it sank. If it was still inflated, it would have been at the surface. It's eventually degraded and filled with water, and then...
Yeah.
It doesn't look that old either. It looks pretty clean. Did we get a Niskin fire? Okay.
Thank you. Clarification.
So we're going to take a moment and collect our eDNA sample.
Yeah, and if you're just joining us and you see a soccer ball on the right-hand side of the screen, it is indeed.
We see it too. Jeez.
Somebody's...
These white rocks.
Yeah, it's really blocky.
1454. Thank you. Yeah, we do have some rocks. Really white.
I'm just confirming the Niskin has not been fired yet,
no,
right?
he said 1454 it went.
Thank you.
Mm-hmm.
And I was
Yep,
thinking about soccer, I'm afraid. That's great. So that is our second water sample from which we are going to extract environmental DNA, which is loose DNA. Comes from loose skin cells from organisms, and also maybe gametes, sperms or eggs that organisms have broadcast into the water. And this will be another tool that allows us to help characterize the biota that's present on the Blake Plateau and the various localities that we've been able to visit.
Ooh, you got a shrimp in the water?
Yes, we do have a nice red shrimp floating by.
These look like calcium carbonate rocks. I'm not a geologist, so I don't really know. Anyone chime in on the chat log about it?
They should be.
I think I see something on the water column, too, at 1 o'clock or 2 o'clock. Looks silvery, maybe a fish in the distance. Yeah, a little silvery guy. There's something there. Too far away right now.
I think you're right, though.
Yeah, it's too turbid. Some kind of cutthroat eel or something.
Good eyes.
Yeah, we were talking about getting a geological sample. Ideally one that's local and hasn't necessarily fallen down from above, but it looks like that's all we have here. But it is in the middle, so maybe we could get...
Exactly. Maybe
one of these
edge.
rocks would be good to get. Yeah, you think? Do you think one of these would be, because it'd be super old? Or do you think it has fallen down from above? It's not attached.
Could have fallen from above, so I think...
we can always collect another one at the edge.
That's true.
I don't expect a lot of fauna here.
Mm-hmm.
So if we do collect a lot, it's going to end up being on that upper rim. So I don't think there's any harm. And it's not like we're using it's a rock box.
Right.
It's like taking up space in a bio bucket. So we can always collect 2 or 3 of them.
Mm-hmm.
Not any harm. Of course, that's from my perspective as a biologist. I feel the opposite if I was a geologist. Are you guys ready to zoom yet? Okay. And if you can zoom in on these brown spots in the sediment, they're probably detrital algae. Yeah, this is a brown gunk. Let's see what we're looking at. Yeah, it looks detritus E. Maybe sargassum. Yeah, that's a piece of sargassum. Yeah, the sargassum naturally grows in the shallow areas closer to shore. Sometimes we see it in the mesophotic zone, but ultimately it breaks off and then becomes part of the Sargasso Sea, which is a whole habitat in of itself on the surface right here. So the algae, makes it habitable for specific types of fish and algae and critters that actually look like the sargassum.
Ooh, we got a
that's
nice,
cool.
beautiful jellyfish.
What is that? He's very dark.
It is very dark. Maybe a Cutthroat type, another Rhopalonematid maybe. So
Bloop.
allied to the jellyfish, we were able to collect it early on in this expedition. I hope we see a few of those. That would be great to pick one up. I can't quite make out its tentacles, but
I think he's too far away.
Yeah.
But water's a little too turbid.
we'll see.
That's hard to
But
say.
hopefully we encounter that again.
Here's a cool fish at 3 in the water column. He's different looking. Yep. He's kind of goofy. Can't
It's
swim very
coming
well.
towards you.
See-through. Neat. He's not very good at swimming. Maybe he's just caught up in some crazy currents down here. That looks like a shrimp. There he is.
Nice.
He's got those clear eyebrows. The swimmerettes. Whoop.
Run away! The fish was a cyclothonia, says Mike Vecchione. The most abundant vertebrate on the planet, with estimated numbers of a quadrillion.
Unfathomable to me.
Yeah. All right, get one of these rock samples.
Yeah. Anything you can grab will work, I think.
Is that an organism?
Yeah, the crinoid and a little crab.
Yeah.
Yeah. Ophiuroid. Yep, the branching thing is dead sargassum that's floated down, so it's
I
detrital.
see.
There's a shell. That's neat. A shell over here right off screen at 3 o'clock, 4 o'clock. It looks like a nice hidey hole.
Great.
So these are about 1 meter tall, white. Let's see, sharp boulders. Whatever you can grab. I don't know if we have a preference.
Looks conveniently situated.
So this is going to be 03G. It's our third sample of the day, first tour water. That's odd. That's a sharp-looking thing at 9. Wonder what that is. This looks like like a nail from like one of my like a train rail nail almost. I doubt that's what it is, but it's got that same kind of shape. This rock is about 15 centimeters wide. If it's not attached, we'll take it. Looks like you got a couple little worms or something growing on.
I think those are little hydroid colonies. So
there's a worm in the sediment.
Look at all the shell hash.
Oh yeah, my goodness. Cool.
Oh, you can see pteropod, many pteropod shells.
Oh, what a cool thing.
So those pteropods are the pelagic snails.
Yeah, there's an amphipod or isopod right in dead center screen. It's white. I can see its black guts. Oh, and then the Galatheid at 3. Sediment sample, maybe? That would be a good idea, too. Maybe get a sediment sample. Hey, is someone call in?
Morning, all. Scott Brents, just calling in.
Hi, Scott.
How are you doing?
Good.
How about you?
Oh, it's a crazy day. Crazy day. I'm looking for it to get better. So, I just want to be sure you're seeing that the geologists are asking for the least eroded and whitest rock you can get.
The leached, eroded, and whitest rock?
All right.
Yeah, the least bioerosion. The more bioerosion.
Yes, I think we should.
The least bioeroded.
We should reassess.
Okay, hang on.
Sorry, we're just getting requests in the. Thank you, pilot. Sorry. So we were looking for whiter pieces, and
this might be it might still be a good spot to grab. There might be. Yeah, do you think they think there's something better over there that's less eroded and more acceptable?
The biggest question, of course, is which one's loose, right?
Yeah, I don't even know. Any of them are loose.
Yeah, exactly. You don't want to pull the plug out of this hole we're in.
Mm-mm.
Well,
it
does look like a
human
piece of
debris or some kind.
rebar or something.
Here's something funny. Maybe some oysters, like a cluster of oysters at 9. Yeah, if you could go full wide and maybe reassess and see if we can get a. This is actually not really eroded. This one.
Geologists are asking. They think that a piece could be broken if it's heavily fractured. So it might be able to knock off a piece.
I'm looking at that white piece that's standing up just behind. There's that big crack right in the center of it.
At noon.
Sort of brownish round one.
Pilots
I think what Jason's saying is you could probably just break off the top of that, perhaps, or the left side of the
more 1 o'clock
fractured there.
Yes. Okay. We don't want to actively break. Yeah, that's crazy. Okay.
Understood. Thanks.
But it looks like there's also some loose stuff over here. Maybe that's not as eroded as the one that we have, or if it's similar. Okay. Thank you. I mean, I think they all look pretty similar. Maybe one here in the middle of this pile is. They're not very eroded in this area at 3 o'clock.
Right.
Maybe that's better. Might be some more.
Yeah.
Yeah.
Essentially, a whiter piece with less pocking, less grunge as possible. There's a little eel in the background there. Not fairly large eel.
Mm-hmm.
Yeah, could we have the lasers to get it again? Oh, there they are. Sorry.
How about this one right in the front?
Yeah, it's sort of square block right in front of the larger piece to 7.
6 o'clock? Yeah, 6, 7 o'clock. Mm-hmm. Will that one work?
Yes.
That's
Thanks.
probably loose.
And then we can make our way to the edge of this wall.
Mm-hmm. I would think so. Like, I wouldn't squeeze it too hard. But it shouldn't just, like, fall apart or anything. We can do it on the surface since we are collecting it.
Mm-hmm.
We can do it when it gets up here. If it is really loose, you'll see it kind of like ooze, if you will, like dust off. All right, so start sample 3.
See how soft this is or isn't.
Yeah, it's loose. Looks like this might be a piece of dead Faria here. This is 6. That's promising for the upper walls. We have a yellow 8, I think 8-armed crinoid at 9. Sand's pretty soft.
And while we're here, do you think it's worth sucking up a little bit of the shell hash?
I mean, I
as
would.
a sample? Yeah. Okay, so this is very brittle, this rock collection.
Yeah, it broke pretty easy. You can see some of the, I don't know if that's loose sediment on top of it or if it's just flaking off the, it's almost like chalk. Mm-hmm. Yeah.
Mm-hmm.
Very chalky. Can we get a sediment sample too while we're sitting here?
We want to target where the largest sediment size is to pick up hash, shell hash.
There should be some pretty good shell hash right next to these rocks, just because the larger pieces have a tendency to collect. Yeah, that's good. Yeah, a little to the right a little bit. See all the little shells.
Yep.
Ostracod shells. This is different. That looks kind of like a paper octopus, right? Or paper nautilus shell a little bit.
Yeah, an octopus.
Yeah, the little lap to the right a bit. You can see all that thick, large shell makeup.
Argonaut was the word we were
Argonaut.
searching for.
Argonaut, yeah.
Argonaut egg case.
So it's going to be 4G. And it's going to go into the suction sampler.
This will goes into canister 5 first, right?
1.
Oh.
Whatever you have is fine.
Yeah,
Just
whatever's.
let us know. I think 1 is our target canister right now.
Good to know.
I'm going to...
So this is classified as a geology sample, but it will also have, no doubt, interstitial fauna. There's quite a lot of life forms that live in and amongst the grains of sand. Yeah, that looks terrific. Thanks. Yeah. Slurp it up. think mostly surface is what we're interested in, yeah.
Little ostracod shells.
Yeah, we should get ostracod shells and all these pteropods and maybe a little argonaut shell.
That argonaut shell.
And I was
Yeah,
thinking,
get that in there.
there's probably quite a number of worms. You know, they're interstitial. Did you know that there are some jellies that live actually in the sand grains?
No, of course I didn't know that. That's cool.
And they are...
There's docile versions? Or,
yeah, so some of them are directly descended from midwater group. You know,
neat.
direct developing midwater group.
I mean, I know Cassiopeia lives upside down on the dirt, but
Yeah.
that's a shallow water photosynthesizing
There's
one.
a whole group that's Halamohydra.
Halamohydra.
You're
Go ahead, Nav.
just hoovering it up. I think that's good. Thank you very much. And that did go into canister 1, correct?
All right, want to make our way to the wall?
Exactly what we want to do.
Yeah, I say we make our way up to that wall, and then... All right, let's check the salinity. Still 35.0, and our temperature's 4.3, so there's no change in salinity here, so I don't see any evidence of like a freshwater outflow. At least not anymore. Maybe there was one some geological time ago.
Mike Vecchione points out that there's a sipunculid that has, as far as he knows, only been reported from pteropod shells. So we'll have to keep an eye out for some worms in there when we get the sample back. Make some biology samples out of that geology sample.
That's end collection 4G.
That's great. Now we'll make our way to the side and hopefully see that nice red jelly again.
There's another one of those yellow crinoids on top of that rock. Otherwise, fairly barren.
Yeah, not full of life for sure. But as you said, our expectations are that the more densely populated part of this hole is likely to be the rim area, just short of the rim and up to the rim.
Yeah. That's pretty typical of a sinkhole. However, this is the first soccer ball I've seen at the bottom of one, so...
This is the first sinkhole I've explored.
The sand's quite smooth here. But yeah, we're in the middle of it. We're right dead set in the middle of it, so you can actually see our location on feed 3. These brown blobs are actually probably sargassum, which is floated down from the surface. A type of algae.
Another great example of transfer of nutrients and carbon to the seafloor.
The sediment's really smooth here, so there's probably not a current in this exact spot.
Yeah,
but back further, when we first landed, it looked like a lot of water movement based on the rippling. Maybe some seagrass or something? Oh, yeah, what is that? Sticking out? Detroit old bamboo coral, dead bamboo coral stick?
Looks dead, yeah.
Yeah, that's
Maybe
promising
fallen,
for the upper rim,
fallen
too.
from above.
It's okay, I can tell what it is.
We got a good view. We're anxious to get to the side.
So we've come up almost, 13 meters
Yep.
from the base of this.
Or about.
Oop, there's something walking around at 3. If you could zoom in on that.
Little crabby.
Maybe a harmy. Oh, he stopped moving.
So you got anemones on them?
Yeah.
Yes. Wonder if this is another
Little decorator guy.
Parapagurus, pagurid.
Again, you can see this sand shell hash is just everywhere. These little teeny tiny shells.
So
Cute.
shaped like a turban. It's nice.
Cute little hat.
Yes. As we were saying the last time we saw that it's a mutualistic relationship. The anemones benefit from the messy feeding of the crab, and the crab benefits by having protection of the anemone and its stinging cells.
That's right. Looks like some more of those white rocks ahead at 1 o'clock. We're facing the southwest wall of this as we get closer. I think the rim of this hole is, 280 meters wide, I believe Derek said. About 68 meters deep. Let me get. Yeah, it looks like the rippling's increased a little bit as we come up the slope. We got a red thing in the water column, or maybe not. Maybe it's on the sediment. Yeah, the red thing.
Take a quick peek.
Yeah, snap zoom. Huh. Not a clue. Looks like a piece of dead something.
Yeah.
Shell or...
Oh. Oh yeah, maybe a piece of shrimp, pelagic or mysid or something.
Could be a
No.
they cast off their, what's that called, molt.
Yeah.
Could be a molt piece.
So Scott France in the chat room informs me that the crabs also sometimes make relationships with zoanthids, a relative of a true anemones, and it's a very similar relationship. And so we might want to go review that footage and.
Yeah, it's hard to tell. The anemone or zoanthid or whatever the nidarium was kind of was tucked. It had its tentacles tucked, so that's different. Rock with a hole in it.
Pockmarked piece of carbonate.
There's some stuff here growing on it, maybe, or have landed. It's just sedimentation on top. Looks like shells or something on the that have fallen. What are those long white shells? The little round ones are ostracods, and then the spiky one at 4 o'clock.
No, I think the round ones are pteropods.
Pteropods.
Yes. But the
spiky one I thought was a pteropod.
Oh. Maybe there's multiple pteropods there.
Sea butterflies. I don't know how many species of sea butterfly there are. Yeah, you can see the increase in the rippling in the sediment, which is indicative of the movement of water above it. Little fish. Ooh, I think we blinded him. Went right into the dirt. Awesome. Sounds good. I say we get to that wall and maybe do a water sample at the base of the wall.
Yeah, I think that's a great idea.
When we get closer to the edge.
Mm-hmm.
Oh, yeah, tripod fish. We haven't seen any of those yet.
Oh, that is
and some shrimps,
great.
too, at 9.
Now we can figure out which one of these.
Ooh. Nice. I believe tripod fish are blind. Is that right?
I'm not sure.
Stand up on their modified dorsal and pec fins. This
looks like, well, I don't know. We have our little fish guide here. Yeah. Bathypterois, though.
Oops.
Question is, what species?
And if they change color or not? It's hard to, fish change color often, so.
Mm-hmm. But it has the shape of this
tail.
dark-headed tripod fish. A little backwash. That was nice footage, though. Certainly enough for
IDs.
Yeah. Bathypterois.
Yeah, sediment tear is really loose. There's something in the water column. Squishy guy. Oh.
Is that
a pteropod?
That is a pteropod.
Nice.
A sea angel.
So that's the shells we're seeing are from this critter.
I think this one is a shellless one.
America.
This one, yeah.
Okay, a relative of this critter?
Yes, the relative of this critter. It's just beautiful. I wonder if Mike Vecchione's in the chat. I wonder if he'll call in. I know he knows quite a bit about these pteropods. Voracious predators.
The jellies or what?
I can't recall off the top of my head. So I'm sitting here. Oh, it's so beautiful.
Mm-hmm. Nice. You can see its orange guts really well on his red tail.
Yeah, so that's...
Hi, who just chimed in?
Hey, Mike Vecchione here.
Hey, Mike, how are you?
Hello.
Oh, can you not hear us?
Yeah, you're going to tell us about the pteropod, this gastropod
Fortunately,
without a
that
shell?
one was easy to identify. Let me mute my my TV. Hang on just a minute. Yeah, that was Cleone limacina.
Cleone.
And yeah, that's one of the naked pteropods. They don't have shells when they're adults. They do have a larval stage that has a shell, but they feed on the other kind of pteropods, the ones that do have shells.
Oh, nice.
And Cleone limacina feeds in particular on the rounded ones. And you were talking about the ones you were seeing in the sediment, they're both spiky and rounded kind. Pteropods come in both of those kind of morphologies, and there's a whole bunch of different genera in the sediment here. So,
oh, nice.
But Cleone is called a sea angel, and it feeds on the sea butterflies.
Thank you.
And it looks like we're coming up on a cusk eel right now.
Yeah.
But what do I know? It's a fish.
I know, right?
I think it has a parasite. Or is that?
Yeah, it does look like
it
a
looks
parasite.
like a parasite on his ventral side. I don't think we've got any fish people in the
Chat today?
in the chat right at the moment. I haven't seen any. Need to get some of those people involved.
So
I can't even tell you for sure what kind of parasite that is. Most of the parasitic copepods have egg sacs dangling off of them. I don't see that on this one.
Yeah, I have no
the
idea.
head, and then also getting a close-up of that projection on the underside of it. It's probably a parasite.
So I called in to talk about the pteropod and not the fish, so I guess I will hang up and
Okay.
Yeah.
step out to get my lunch.
Thank you.
So those
I'll be back in a
pteropods
few
are
minutes.
essentially they're kind of mollusks. So at the family level, that's where they fit in, and they are gastropods, a class Gastropoda, essentially a
the
swimming slug
pteropod.
without a
Actually,
shell.
Since I'm still on, I'll tell you that the two groups, the gymnosomes, which are the naked pteropods, and the thecosomes, which are the shelled pteropods, were originally thought to be related, and then somebody looked at them and said, wow, those things really look very different. So they were separated and considered not to be closely related until somebody did molecular work and found, yeah, they really are sister groups. So the group Pteropoda has been resurrected as a result of that.
Oh, nice. Thank you.
Okay, now I'm going to hang up.
Okay. Enjoy
you,
your
Mike.
lunch. It looks like we're coming up on the wall, maybe?
Yeah, there we have
The
it.
base of the wall?
So this may be a real outcrop of the
Yeah.
limestone in this plateau.
Yeah, you can see the layering in it as we get closer.
And
That's pretty cool.
this would be someplace I think we'd be looking for an additional rock sample
And a water sample.
and a water sample, exactly.
Still, the salinity's still exactly 35.0. Hasn't changed at all. Possibly expecting some, like, freshwater seepage that could have caused this, but it looks like it's a lot older than that, maybe. Look at the neat layering in the rocks.
It's great.
Yeah. If we can get a water sample when you guys are ready, that would be great.
Should be Niskin 3, I believe.
So that's going to be 5W, right?
That's what I've got.
Okay.
I'm scanning around here looking for a
There's a pretty white
small
one.
rock.
Yeah, what about these right here? They're not really eroded, are they?
The ones that have fallen off? I think they...
Look what, something attached.
Or,
I don't know if we can get something attached.
I know, that's what I'm... See what the geologists have to say inside the chat.
Ideally one that looks like maybe it's old.
well,
they still want white, uneroded.
Yeah.
White, uneroded.
Or just broken off, something that they can then know that came from this place.
Looks,
right
looks
off
similar to the wall.
Yeah.
How about this? Yeah, maybe on top of that ledge. Yeah.
Mm-hmm.
How about this one? This black one right here.
Yeah.
At 6. Yeah.
No, just to the left, I think. Yeah.
That little one right there is what I was eyeing, but
yeah.
Yeah,
yeah,
dead
right
center.
there.
Any feedback from the chat?
Just that the displaced material is hard to know where it actually came from.
Yeah.
That's a good question. Sorry.
How big of a sample can you get on a rocks? Can you pick up that thing? Okay.
No.
Yeah, the black one right there is too big. Okay. How about the one by the fish that we had eyed before?
I think
that
that's
looks
a
like
good...
it's local. Oh, maybe we could do that then.
Yeah, let's try that.
Looks like a similar color to the wall surrounding it, so my guess would be it's from there. Yeah, that. Can we get that one? Try. Yeah. Yeah, the other one that you looked at before looks more realistic. These all look pretty. Oh, there's a neat, maybe a cauterized.
Ooh.
Yeah, that guy. Yeah.
Thank you, pilot. And we can potentially get another rock, correct?
I think so. There's plenty of room. No, I don't see any harm in putting this in the same rock box because it's really obvious that's not the same. We can tell the difference. If the geologists are okay with us mixing in the... I mean, we're not taking DNA from them, so...
True.
And then there's even more room.
Mm-hmm.
So put one on the other side. Oh, Jason says don't mix them. Okay. So there goes that idea. There's a worm tube on it. If not, we can find another one.
So we'll see if we can take a peek at that potential cladorhizid after the collection.
That should have been over this way.
Yeah, I think it was just to the right of there.
Okay.
Who's called it? Ah, I see. Never mind. That was someone leaving the the call line. That's okay. That sounds great. Get what we can. It's great. Yes. We're happy with what we've got now. Thank you. So we were curious if you could scan the rocks to the right, looking for a small spiky sponge that we may have seen. Yes, to the right, and we're looking for a small spiked object or organism that has spikes, a little off-white. And I can't remember the exact position of it, so yeah. Certainly. Perhaps 5 o'clock, a little white. And now about 6, yeah, it looks like you're straight over it. Yeah, I'm not seeing it in that. I think it's 6 o'clock of where you're at right now, sort of. I think we better let it go rather. Oh yeah, right there on that rock. What do we see there? Yep, right on the dark rock. Exactly. I think you found it. Maybe this is an urchin. Not sure. Oh yeah, I think that's an echinoderm. A sea urchin, unusual to me. That is great footage of it. I'm kind of curious if that's a collect. I want to quickly ask the chat. Yeah, we've gotten lots of comments about it's a weird thing from echinoderm people. Is it possible to pull that in? The suction sampler? There it is.
I also see a piece of coral
Oh, yeah.
right next to that rock. So we ended up getting the rock 6?
Yes, rock 6 was successful.
Do you know where that went? Okay, thank you. I have to scrape the lip of it.
This is delicate work for sure.
Mm-hmm.
Come on, fella.
Yeah, you might have to scrape him. The edge of your suction hose. He's got a grip on there, huh? Here we go, here we go. It looks like he's loosening up a little bit. There he goes.
Yay!
We're in
Wow.
bucket 2. Nice.
That was really nice.
Thank you.
Work. You know, the program has not collected that type of urchin, that urchin group.
Do we have a name for it?
We had a guess of a name.
Okay.
That was Habrosidaris.
Nice.
But it's sort of a question mark. And that was in Niskin 2, I believe.
No.
Where'd it go?
Sample bucket 2? Suction sampler canister.
Oh, yes.
Yeah, not water.
Thank you. Brain malfunction.
Time for lunch. That's all. Blood sugar drop.
Yeah. Wow, that is super cool. And
Yeah, if we can make our way up the wall.
good
That's
idea.
my priorities. Like seeing a little piece of coral rubble is promising. Same with that bamboo we saw earlier. All right, we're going to make our way up the wall now. So if you're just joining us, we are 186 miles from Florida along the Blake Plateau, diving in a blue hole or a sinkhole. Seems to be the only one out here. We have a lot of multi-beam in the area, and this is the only sinkhole that we've seen in that multi-beam. The top of the sinkhole is at 1,146 meters, and the base is at 1,213, and we landed at the very center, the beginning of the dive. At the base of this sinkhole was mostly sediment with some of these white rocks, which we collected a rock, and that rock was really soft, almost chalk-like. We're going to head up the wall now after our pilot changes. It's definitely lunchtime, so everyone's shuffling. The highest slope in this sinkhole wall is about 52 degrees, and the total overall size of the mouth of the sinkhole is 280 meters wide. So we're going to start, we start at the bottom, we're going to make our way up to the top edge. There's very little fauna down here at the base. That's pretty typical. Sinkholes, the few sinkholes I've been in, and John Reed, my boss, was saying there's not much fauna at the base. There's more along the top rim, and we did see a dead piece of hard coral and a dead bamboo coral, so that's promising for the likelihood of there being fauna at the top. But right now we're focused on geology and picking up some rocks, so maybe our geologists can age them at some point. Yeah, so that's the update. So just be patient. We're switching pilots, and then we're going to start making our way up this wall. Overhangs. Look out for, we see a lot of, not out here necessarily, but a lot of fishing line are the ones that are closer to shore. So just keep an eye out for that too. And anything falling off, because these rocks definitely fall from the top. It looks like another cutthroat eel or something, dead center. There's a few of them, actually. And the sediment's pretty thick. You can see the waves, which is showing the direction of the current near the base or the bottom. For the most part, I just see these white chalk-like rocks. Some layering. Maybe some outcrop here. What we call scintillation holes, which is the holes that are caused by water movement. That's what that looks like. Maybe there was some tunneling here at some point. Maybe you have something at noon. It's weird looking. I think that might just be rock. Nice little cave. Is these, what is that? Can I see that thing at 9? Yeah, it looks like an anemone or something. Yep. Yeah, it looks like a little anemone. Or a corallimorph. Thanks, guys. Yeah, it looks like a corallimorph to me. Yeah, we're about 40 meters below the surface of the edge of this sinkhole. So hopefully when we get to the top, we'll see a lot more fauna. This is a pretty cool geology dive. You can see how eroded the white rocks are, the wall of this sinkhole. And the sedimentation likely coming from above. I see a black coral, dead center screen. A little whip coral, Stichopathes of some type. Oh, no, don't worry. Just keep going. I want to get to the top, especially if we got weather. So Nolan's saying, good. Oh, there's a neat fish. We got something, a fish at now 4 o'clock. Really flathead. I believe we saw this shark-like fish on dive 1. I believe that's a type of shark. Can tell by his tail. Yes, Mike Vecchione said is his shark. So looking at the wall of this, I see mostly this black layered type of rock, and then some exposed white parts. It looks like maybe some of those are the fresh fallen areas, and their pieces have migrated to the bottom of the sinkhole. About 30 meters below, there's another black coral, likely a Stichopathes of some kind at noon. Ooh, that looks like a Chrysogorgid. Right in dead center screen. Can we get a zoom on that if you guys feel safe? Thank you. Yeah, it looks like a Chrysogorgid to me. A little amphipod or isopod living in it. Or shrimp, maybe. Looks like a shrimp. There's a blue sponge in the back. Some crinoids. Nice. It's just hanging out on the wall. It seems to be a pretty good current flow down here based on what the pilots are saying about the movement of the water when we arrived, and also the rippling in the sediment at the base. Light right here? Okay. Yeah, we're good. Thanks. Me? I'm good with it. Oh, I think there's some trash, a can at 2 o'clock. Something stuck in the wall there. Yeah, it's weird. Can you zoom in a little bit? Yeah. Okay. Yeah, it looks like it's right coming into screen now on the upper right. Looks like a tin can. A beer, maybe? Root beer. It's a root beer. Nice.
Did you just say nice?
Yeah. That's terrible. Recycle, everybody. Reduce, reuse, recycle, and don't throw your trash in the ocean, please. Yeah, it looks like the wall's kind of breaking up a bit.
So did I miss a shark?
You did. You missed the shark.
Was it a cat shark?
No, was the same like long-nosed one we saw on the first dive? A really long-faced shark. It looked similar to that. We had a name for it.
Flat-headed shark.
Yeah, we had a name for it when I redid the notes after I was looking through emails and stuff.
Mm-hmm. So we are at 1172, and the rim is at 1146.
About 30 meters away. Yeah, Sirius View has an awesome shot, actually, of D2 in front of this wall. You can see how, look at the wall formation and the layering, and even maybe places where the wall has broken away. Which you can also see in the main screen, where you have the white and the black, with the parts that are white and parts with black coating. So the parts that are white have likely just been exposed more recently. Not just, but geologically just. Nice little bioturbated hole there. Looks like somebody might be living in it. Based on the sand.
This is a rind on the right.
Like the old that's been there for a while, right? Versus white, which looks like it's maybe just recently
Sloughed off.
sloughed off. That's pretty cool. Wow, that wall is straight up and down, like 90 degrees almost.
That is stark.
Yep.
Thank you. Oh, there goes a little.
Something in the water column. Is out of focus? I don't know what it is.
I think it's a Medusa.
Could
Well,
be.
that's actually longer.
Yeah, you can see the water is dead still here. It's not moving at all. Can you look at the wall at 9 o'clock? I see some yellow stuff and some red stuff. Some biota.
Oh, yeah.
Yeah, right? Yeah. It's like now it's just center screen. Little brown guy. Yep, dead center. Maybe Feria? Looks like some sponges, maybe? And Antipatharean. So that's, oh yeah, look at that. That's gorgeous. There's actually 2 species of Antipathareans and then at least 2 species of sponge.
Wow.
It's
It's
like
just
Feria-like.
out of nowhere, these.
Maybe even a solitary cup coral. And I see a crinoid. Yeah, that's fine.
Yeah, so this does look like a farreid
Yeah,
sponge
yep.
in the background. We'll get a look here
This is
at this black coral.
Antipatharean in the front. Very fine, feathery. And then if we could, oh, there's even an anemone or something on the wall here.
Yeah, you can see the
See the
tips of its
tentacles?
tentacles.
And then at least another species of
I
sponge,
think
the
there's a
yellow.
little hermit right in there.
Nice.
This is like a little oasis on this
All
wall.
right. Think that yellow one is a peripheron? I mean a demo?
I think that, yeah, the larger one may be a demo, but the other has that Hertwiggia yellow look to it, the yellow Hertwiggia look.
The crinoid. And a solitary cup coral, it looks like, at the top.
Oh, and here's I think a shrimp there, the 10 o'clock.
Nice.
Ooh.
We can quick zoom on this purple one and the stuff behind it, and then we're good. Yeah, it looks like a. Oh, there's even a squat lobster on there. Sweet. Nice. Squat lobster on an Antipatharean. So this is a different species. I'm not sure either of these species. They're both, this one's more of a maroon color, differently shaped. There's also some worm tubes in the back.
Yeah, and some more polyps, some tall, thin polyps.
Solitary cup corals, maybe. We're good whenever you guys are. Yeah, there's solitary cup coral in the back, yeah, on the rocks.
Cutthroat eel was spotted as well.
Nice. These look like Anthomastus, possibly. Maybe.
Whoa. Yeah. Definitely octocorals, Anthomastus-like, with really long, extended polyps.
Yep, we're good whenever you are. It looks like a dead solitary cup coral to it, right next to them at 9 o'clock. He's blended in with the black background. He's been there a long time. No, we're good. Yeah, you can see here this rock, just look at it. It just looks like it fell right off.
It sure does.
Square block.
What's that in the water column?
I'm not sure. Little guy. There we go. Jumbo pod rocks on top. Some more Sticopathes or black coral stalks.
We're about 25 meters from the top.
Can we get a snap zoom to that center screen? I see a gorgonian of some kind. Yep. I'm not sure what kind. Maybe a primnoid of some type. That's ophiuroids. Very fine branching, planar, long branches.
Again, you can see this diversity of small encrusting sponges.
And some crinoids.
Parantipes.
Parantipathes. Yeah, we're good, thank you.
That must have been one of the two Antipatherians from down below, but I'm not sure which one.
Yeah, I'll Google it at the end and make sure my notes are right. Do our corrections. Niskin sample, yeah. Ideally, I think at the top of the rim, or do you want to do center?
At the top of the rim, and then we'll do another one,
At
before we
end.
leave.
Yeah. Can we snap zoom underneath that cave? Something funky in there. Dead center.
Now you can see we're really getting a lot more.
This is Detrital.
Yeah.
Detritus of Sargassum.
But out on the outside of this.
You see a lot more gorgonians popping up. A little increase in life.
Well, I stepped away to eat. Did we make any collections?
No.
Thank you.
We're trying to get to the top.
We're making it. 1,169 meters.
We're going to 1146. Oop, we might have missed some deep sea predation. I thought I saw a fish eating something.
Oh.
But it's gone now. Oh, or we're freaking him out. Hello. There's a nice gorgonian coming up off screen. And another anemone
huge
dead center.
fly trap anemone.
Mm-hmm.
This looks like the richest zone.
Yeah, so far there's lots of fauna here.
That fan is huge.
Yeah. Can we zoom in on that fan, if that's okay? Yep. Yeah, center 1 o'clock. Yeah, that guy. Nice. So...
Bamboo.
Bamboo coral, yep. Nodal branching,
And
planer.
so that's what the nodal branching means when this, I see that
The branch
very
comes
clearly.
off the brown ring.
This one has relatively... The polyps don't obscure everything else. There's some crinoids on this bamboo coral.
Looks like it's flattening out a little bit here. It's almost like a stepping, right? Like it steps down. Steps, yeah. Lots of sedimentation, likely from the upper surface. We got some kind of solitary stick-like dead center. Might be a dead thing or...
Curious what's floating by there in the foreground.
Yeah, I don't know. Lots of cutthroat eels are pretty common. You can see the steepness of the wall here. 25 degrees maybe. 15 degrees. But then that 90-degree angle wall in the background. Yeah, current's really still near the wall. Really hardly any movement at all. Yeah, we were. Okay. Yeah, so you can see that stepping. It's
Oh.
really neat.
Wow.
Oh, man. Yeah, that's straight down.
Now it's a nice view of the edge of the sinkhole.
Yeah, it's 90 degrees, even maybe more of that. You know, have those in cuts, outcrop in cuts. And the layering again in the geology, you can see the rock layers. That's pretty cool. A little bit of rippling here in the sediment.
Yeah.
Ooh, TN4.
Yep. So I don't know. So I wasn't sure I was expecting such a stark rock rock outcrops on the side.
Yeah, it's just rock wall. What's that red thing?
I don't know. Can we zoom on that red thing? Yeah.
Yep.
Thank you.
Shrimp. Shrimp.
Yeah.
Bright red shrimp. Those little swimmerets going crazy.
Thank you very much. Perfect.
Get more of these black rocks.
More pelagic organisms, a little ctenophore floating by. Maybe a medusa. A little.
You can see how I would call this area kind of active geologically, right? With all the white exposed, freshly fallen, versus the black pieces or the encrusted ones. That ferromanganese they were saying.
Right.
So it's more recently exposed than the. Now, granted, to get that black coating is a couple million years. Is that right? So.
Very slow process.
When
but
I'm
still.
talking about active, I mean in geologic terms. There's this really white area here at the bottom at 6.
Just looks like
Okay. Okay. Okay, no worries. There looks like some type of biota at, look at the layers on this. That's super neat. At 3 o'clock. Yep. Yes. It's just like a light pinky color. Looks smushy. Probably a chrysogorgid from this distance, but. Oh, no, not a chrysogorgid. Some kind of gorgonian. Flat, planar, sparsely, well, it's pretty heftily branched. Maybe bamboo coral on the bottom one? This one down here. That looks like that's bamboo coral, that one. Yeah, it's got multiple branches. Looks like the bottom half is dead. Nice.
really nice.
You can see the pock marks on the rock walls when we get close. Yeah, there's a big white patch down there.
Yeah, some more freshly eroded away. I just can't. This reminds me of limestone cliffs.
Yeah, like the cliffs of Dover, right?
When you drive around and.
So that looks freshly exposed. There's even detritus in it, like the Thalassia seagrass. Really smooth. There's a little teeny tiny fish fry. Cool.
As the ship maneuvers up above, we'll be able to get in a little closer in a moment.
Yeah, we're getting some weather coming in. The boat was rocking around a little bit worse than, it was getting rough.
I'm feeling it.
That rock up there looks like it's about to tumble off. It's like hanging on by a thread.
Perched.
Yep. Yeah, let's not, hit that guy.
Yeah, thank you.
Floaty guy. Oh, schmutz.
Yeah. Little marine snow.
Oh, there's schmutz everywhere.
Raining down from above.
There's another shrimp at 9. Maybe the same one we saw earlier. I don't know.
Another cutthroat eel to take a look at.
Oop. Oop. Sorry, buddy. We're probably blinding him.
He's thinking, who put that there?
Whoa. Yeah, I haven't seen no change in temperature or salinity. So probably this formed during like the last ice age or something, where there's more exposed to the surface. That's a guess, huh? Let's see.
It's a nice looking sponge.
Yeah, that is odd, isn't it? We saw something like that yesterday. It was clear. Okay. Thanks.
It's great.
So this is kind of what it looks like in the cave diving in Florida, with like that really white, really white light colored walls. I've only seen pictures. I don't think I could bring myself to do that. But it's terrifying. But there are people who love that. And I'm sure if I could handle it, I would think it was cool, but definitely can't. This white piece looks interesting. Right here. Like, that's pretty fresh. The one at 3. Yeah. Yeah, like it came from this little chunk here. I'll pick it up and put it back. Yeah. That's really light colored. Yeah. Smooth. Not a lot of pock marks.
No, it'd be interesting to... We don't have... Can we put a geological sample in one of the bio boxes?
I'm fine with it. I don't think that will fit. I think that's humongous.
Oh, no, I know that. But I was wondering if something that we know that is pretty freshly come off might be something that the geologists would like to have. We'll see if they have anything to say in the chat.
That looks like another piece there. That looks fresh. This chunk right at 9.
Just above the eel?
Yeah, it looks fresh. Yeah, let's go slope. Sounds good. So the top of this feature is 1,146, and we're at 1,167. So we're about 20 meters from the top. 20 meters deeper.
There we are, getting a look up.
It's broken pieces
It's
piled up.
Interesting not seeing... I sort of expected more life here, but it seemed like that it was more dense, a little bit deeper in.
Yeah. Can we look at these holes at 9? Installation holes or something? It has a pretty different texture below that like line, right? It's like, no, they're not holes. They're something living on there, maybe. Yeah, discolored sponge, maybe black sponge. That's neat.
I think so. More encrusted.
An old foothold of a coral.
Oh, an old Because you see the central area,
yeah.
maybe they're hold fast of large organisms. But you see the small demo sponges to the left. Good one,
Thank you.
Roland.
Yeah, we're good.
Yep,
Good. Yeah.
thank you. One of those hold fast seemed to have a sponge growing right on top of it.
Look at this jumbled pile of rocks.
Then we have
It's like a bamboo coral sticking off, too.
I think it may have an associate that's climbed up on it, maybe.
Yeah. At like 10 o'clock.
Yeah.
Something maybe living between the rocks. Yeah. Maybe just detritus. Yeah, detrital. Some rope.
Mm-hmm.
Okay, so that means there's been fishermen close by or fishing gear close by. Oh. And then it looks like two different species of gorgonian in front of us. They seem to be similar to the two we saw earlier. One is a bamboo coral. And the other one looks primnoidish. Yes, you can see the bamboo coral. It might even be a bamboo coral, too. I don't see any striping, though. Yeah, the bamboo and the primnoid next to each other.
It's interesting.
No, you can keep going. Thanks.
It looks like exactly what we saw down below.
Mm-hmm. Yeah, like
those
right
two
next
right
to
in
each other.
association.
Mm-hmm. So steepness kind of settles out a little as we get higher up, but the rock jumbling increases. What is that dark stuff?
That fits with the contour lines we're seeing.
Mm-hmm. Yep, but steepness decrease as we increase the height on the wall.
Right, get up to the lip here.
This looks like it could be a trash bag, but I hope it's not. No, wow, I don't know what's going on here. What do you guys think? Just old rock? Mm-hmm. That detritus stuff at 9. That's that's really old dead sargassum.
Yeah.
Maybe and some oh my goodness, seagrass. Stichopathes, orange stichopathes. Another gorgonian at 9. I'm sorry, noon. Stichopathes at noon.
Hey guys, how's the dive going?
Good, Matt. How are you?
Looks like a really cool wall, man. We got lucky today, huh? Fun. So, the weather is building on the surface for everyone at home. So, we are looking at an early recovery. We're gonna make the call shortly when that exactly will be. But just to let everyone at home know that we're not going to be down here for the full dive time. That we're going to have to come up early with an abundance of caution for our people and our ROVs. So, yeah, I'm sorry that we have to leave such a neat site. It's been really exciting, but just a heads up. And Steph and Alan will let you know exactly when the pilots say that will be.
Thanks, Matt. Can you swing 9 o'clock? There's some yellow stuff. I think it might be human debris. Yeah, some human debris.
Oh, unfortunate.
Keep your trash on your boat.
And blue behind?
Yeah. Oh, it looks like a
that's-
balloon ribbon. Got the cool blue sponge too. Yeah, deflated balloon ribbon.
I say boo to that balloon.
I know, right? Your dad jokes are going to kill me.
I was just booing. It wasn't meant to be a dad joke.
I don't think you can help it. There's a cool single-stalked furry thing at 3. Yep. 3, yep. There you go, dead center. Yep, furry guy. That looks like a zoanthids or worms. Hey, who chimed in?
Hi,
Maybe.
Stephanie, Scott France, back from my lecture.
Hi, Scott. Yeah, I don't know what I'm looking at. It looks like an infested stalk of some kind, maybe zoanthids or worm tubes.
Can we,
See what I mean? Like,
can
it
we
looks
see
like
the
worm
crown?
tubes?
Yeah. Just like I would like to see the crown of these, the tentacle crowns.
Maybe they're hydroids?
Yeah, I also would like to see this lit up a little bit more. I wonder if it's cornularid. There are some octocorals that have an external tube, kind of like the perisarc of hydroids, Alan.
Oh, okay. I think it may not be in the cards.
Whoa, that's a big bamboo coral.
I think we're headed in that direction.
Yeah, that'd
Yes,
be
please.
good. Yep, definite bamboo coral. The internodal branching.
I also noticed the black coral behind it.
Yeah.
One of those orange sticopathes, I think.
Yeah, that's what I thought it was too.
I can see the sclerites through the tissue on some of these polyps. You know, you can see the bright white through the tissue. I don't see the strong needles protruding. That's a good view. Oh, they're there. They're sticking up a little bit.
Yeah, the little, like, lumpy bumpies.
Yeah.
The lumpy bumpies, I'm not sure what that is. That's a little bit different. You mean on the polyps?
Yeah, no, at like the base of the polyps, there's little, like pimples.
Yeah, and they're not on all of them. They're only in some places. We see this occasionally, and sometimes they're indicative of bundles of nematocysts, which suggests that maybe something was there bothering the coral in the past. Great view of the sclerites in the body wall. It's going to be very informative later. But anyway, yeah, so I think those bumpies are not always present, so it's good when we see them.
Neat.
Thanks for the close-up.
Fantastic. I think Chris Ma mentioned there's a star nearby to the lower left.
Lower left.
Well, if I were a coral, that would raise goosebumps on my flesh. Feeding sea star nearby.
Oh no, what have I started?
I know.
What have you started?
Yeah, he was saying lower left, but I'm not seeing it.
Nor am I. It was pointed out in that chat.
Yeah, they're usually landside. They're usually a little bit of a delay between us and them, so we may have blown past it like that.
Yeah.
That looks like sponge.
Another beautiful encrusting
Yeah.
demo sponge.
That's very veiny.
Yeah, you can see the canal system really nicely in this one. Starts off as really tiny canals and then goes through these chambers where they feed, and then they get into ever larger and larger canals and then blown out the oscule.
Awesome.
Alan, is there any known pattern about why you would see so many relatively large oscula on some of these sponges versus others? I mean, that one, it's so apparent.
Not that I know of, because there are so many different patterns and different organisms, and then create a different, flow regime.
I see that patterning, though, on multiple species, right?
Mm-hmm.
We have, like, Macali, and there's a few others that do that.
Yeah, I mean, there's some general principles. Oh, here's this same...
Can we get a
it
zoom
would
on
be great
that
to get a
stick?
zoom on this.
This we looked at earlier. I thought maybe it was, like, encrusting hydroids or
Now,
something, but
is
Scott
this something
said...
that's a collect target, you think? I'm not sure
I
if
think
it's
that's
kind of big, huh?
Go
It is a collect target if it's the sort of corneal area we think, but it requires we're getting the zoom now.
Yeah.
With you.
Well, now we're getting a good look
Yeah,
at
now
it.
it might even be, yeah.
I think it does. It's exactly
Optical.
like he said.
Target. If you could sample.
We need this?
Yes, if we could collect this, or part of this. Wonderful. So you can see what Scott was saying, that it has these like harder tubes, and the each of the polyps is coming out from the tube.
Yeah, it's interesting because we're used to the octocorals having an internal skeleton, and so this is almost like an external skeleton, at least the base of it.
Yeah, it's really weird.
Looks like we're going to get a feather duster worm as well if we get the top of this.
I noticed, I think there was a Gallophian crab on there as well. Al, I'm sure there's going to be some hydroids on the top of that for you as well.
I'll be counting on it.
This is going to be 8B, right? Are we on the same page here? Yes.
Correct.
8B. Okay. Scott, do you have a name for this?
Yeah, stand by. I was just looking up the family name spelling for you. I'll put it in chat, but I think it's
Yeah, we need the living part. So if you can get see where it gets bushy. Yeah, we need at least a chunk of the bushy part.
Yeah. So if it was cut in half.
Yeah, in half would be fine.
Okay, we've got a name, Cornularidae.
Thank you.
It's the family name. That's nice when the octocoral experts are, someone like Scott who's been here so many times, or been to the deep so many times, is identifying something to the family.
And this is sample 8B, 08B. This is probably going to be pretty stiff, so you might need to use your clippers to snap it.
I love watching the collects.
That's good. Yeah. Mm-hmm. That's good.
Now it's free to grow some more.
Mm-hmm. Just want a couple of those polyps. This is, yes, sample 8B. Yes, those polyps at the top is what we were after. Oh, there's a little
Oh yeah?
critter on there.
We've got some hydroids. That's going to be a fun one to play with.
Separate all the associates out.
Mm-hmm. Well, I've had a few easy days, so.
I know. I think it's going to get rough out here too, so we might not be diving too much. They haven't made that call yet though, so.
Do you know the destination box for this yet?
Mm-mm. Yeah, no worries.
All right. Port inner. And success. Thank you. Well done.
Yeah, thanks. And if we can try to get to the rim of this before we have to pull the ROVs, that would be ideal. Looking underneath a little cave. Rockception.
Nice little shrimpy.
Another bamboo, maybe? A couple of them.
We can get up here, we can get a water sample.
We're only about 10 meters below the upper rim.
Yeah, 5-10 meters. We'll be hoping to get another water sample at the rim, right?
Yeah. Yeah, I'm not sure how much bottom time we have left. And so the rocks are much smaller up here. More cobbly sized than they are boulder sized. Some pock holes.
Yeah, you can see where pieces have fallen out, eroded out.
That is what, Metallogorgia? That's the first one we've seen this dive. Yeah, just a quick one though. I know what it is from a distance. Yep, Metallogorgia with its spaghetti
Brittle.
sea star and brittle star. Yep, we're good whenever you are. Yep.
How do they find each other?
I don't know. It's
So
crazy.
cool.
Probably through the chemical sensors, I guess.
My guess is there's not high success rate for the larvae of the brittle stars, too.
Yeah, well, that's true. They are selected
At the-
species, right? Remember that going back to ecology in college? Are selected species where they have like billions of offspring, and maybe one or two will land somewhere. Most of it gets
That was
eaten
the
in the water
concept.
column, right? Yes, lots of sand between here. Maybe what, a 45-degree slope, or not quite that high, maybe. It's hard to tell from this angle. I'd have to turn the camera and look down the hill.
It's a dead colony.
These rocks are really white too, compared to some parts of the wall which are quite dark. So I wonder if the sedimentation's coming off or something to keep them from collecting that for a manganese crust. They're not exposed to the water long enough.
Yeah.
Buried. So it looks like Sirius is at the top of the rim of this feature, but D2's still hanging out on the wall. Yeah. Okay.
I think maybe we could take this opportunity to get the fourth water sample now, if that's...
Gonna do that. Okay. That's gonna be sample number 9. Ooh. Looks like a Demo sponge.
Yeah?
A blue Demo sponge too. A blue one and a clear one.
Yeah, with a shrimp on top. So we have a couple different demo sponges encrusting this rock. Actually, there's a green one there too, greenish yellow. So we made our 9th sample of this dive. Nice eel, pink. see any more of these pink guys?
That looks like the top rim. Look up there.
Oh, yeah.
A really stark edge
Mm-hmm.
right on the distance. Something funky in there. Maybe a fish or something? No, what is that? Yeah. It's pretty
like
old.
Looks like a stump, but...
Stump.
Man-made detritus.
Yeah.
Human debris.
Wishful thinking.
See, I can see the top rim of this sinkhole we just came out of.
Mm-hmm.
There's a couple of Metallogorgias, a few fans, a stump, or whatever this thing is.
Bucket.
Bucket, upside down bucket.
Yeah.
Can see the sedimentation. It'd be cool to look over the edge too and see what's on the other side. We don't really. Oh, okay. I don't see a rise and then a dip on the outside in the multi-beam. So I think it probably just is flat on the top.
The hole really is a mystery.
Mm-hmm.
You know, whatever formed it, it's not happened commonly because this is the only one out in all this space. Want more zoom in on that?
Oh, we can, yeah. 6 o'clock. Yeah, 7 o'clock. Little pink gorgonian. Yep.
Mm-hmm.
Bamboo coral with a crinoid on it. Very planar. Got nodal branching.
And more great, another shrimp there, and great sponges encrusting.
Something with tentacles underneath it. There's a couple of sibillid worms, worm tubes. That might be a sibillid worm tube, a feather duster, right in front of us.
I think we got one on that last collect. And you can see many more pteropod shells.
The sediment. Pretty cool.
Not sure of the genus on this bamboo coral, but I'm going to guess it's in the J clade because of the thickness of the skeleton, the relative thickness of the tissue, and that nodal branching that you pointed out, Stephanie.
Thank you. Okay.
Great.
Do you think we'll have time to kind of go around the top rim a little bit, or are we going to get pulled? Okay. Okay, sounds good.
Just
These rocks are like 10, 20 centimeters wide in this area, mostly sedimentation. I'd call this mostly sediment. Maybe 50% rock cover.
mm-hmm.
And then it flattens out at the top, which we fully expected based on the multi-beam. So this is probably what most of the... Ooh, I see a fish at 3. yeah, most of the bottom of the Lake Plateau looks like. Of course, we're only seeing a teeny tiny part of it, but yeah, little fishy, rat tail of some kind.
And we just got a great recap of the fish we've seen.
Oh, good.
From Ken.
Yep, sounds great to me.
Sounds excellent.
We've got a clump of dark, likely detritus, just from what I'm seeing this far away. Still pretty far though, so it's hard to tell what it is. And then you can see the edge of the sinkhole sloping down to the left, to the right. Yeah.
It most likely did start up there. Roll down the hill.
Mm-hmm. I'm going to do a water sample up here.
I definitely would like to do a water sample, maybe right as we're leaving.
Okay, that's our last one. Okay. Yeah.
Mm-hmm.
Yep. Right before we take off, that would be good.
Oh, we should keep an eye out for the burrows up here. Ken
Like
Sulak
townfish?
No, is commenting that they're the ghostly white blind lobster.
Oh, yeah.
Canthocaris
Canthocaris.
can be in these burrows.
Mm-hmm. I think there's a star at 9 o'clock. Yeah, the one dead center.
Yeah.
There's also bamboo coral coming up at 3 o'clock. Huh. Actually, I see another coral going off screen, too. It looks a little yellow. Let's take a peek at this star. Looks like he's eating something. There's not much around.
Evidently enough, though.
Mm-hmm.
I wonder what it is eating.
It's about 15 centimeters. And the type of goniaster. Yep, looks like a primnoid under there or something.
Yeah, really delicate primnoid, huh?
Mm-hmm. And then on the right,
I
you
think
can.
it's actually a chrysogorgia.
Chrysogorgia.
Gold skeleton.
Oh, yeah.
Delicate polyps.
Thank you. There's a yellow one next to it, too. And then you can see the really good shot of the detrital sargassum that's floated down from the surface. Look at this pile over here in the sediment.
Yeah.
Hey, who's calling?
Chris
Hey,
Moss.
Chris. What do we got?
So this is one of the more common, if you will, goniasterids, and as you noted earlier, it's feeding. This is Circeaster. Circe, as in the goddess of magic. So as you can see, it's feeding on some kind of, I'm not sure, is it a coral or a hydroid, but you can see the stalk coming off of it. These are, after the evilplasmomas and the hipposterias, they're one of the most commonly encountered cnidarian predators. This is the same species that Scott France observed feeding on that Paragorgia a few years ago, the one that we saw feeding on the same Paragorgia a year later.
Oh, my goodness.
These actually figure pretty... I'm sorry?
The same exact stars eating the same exact Gorgonian?
Yeah.
That's crazy.
Yeah, so apparently they saw a Paragorgia that had been downed, and there were 2 or 3 of these Cerciaster feeding on it. And they came back to that exact fallen colony a year later, and there were still those same 2 or 3 animals feeding on it. And
my goodness.
So when you consider the rate of interaction, if you will, at this depth, the physiology doesn't tend to lend itself to very fast things, and sea stars aren't exactly speed demons to begin with. So, if that sea star was feeding on that cluster of stuff, months to even a year from now, it wouldn't even surprise me. It might even be moving on to that. There was a primnoid or some, not a primnoid, but some kind of Gorgonian just off to the side there. So I would say that's next.
Yeah, right.
So, but any of those smaller clusters would probably be just as, they'd be easier to attack. So, but thank you for spotting it and for documenting that. Watching the disc inflate and all that stuff. That stuff we didn't realize was actually like part of their living appearance. A lot of times you get them like that, and people mistake them for actual character features. So looks like some kind of astroschema, a snake star. There's a whole bunch of these, and they're let's just say complicated is
right.
That's that.
The best way to put it.
On the yellow Paramyricea.
Thanks.
Yeah. Have a good day.
You too. And the Anthomastus too, dead center.
How could we not have a good day?
I'm sorry, what?
We can move.
Yeah. Most of these brown spots are detroital sargassum. It looks like a Stichopathes with Goro.
So we have made it to the top of our sinkhole exploration today, and now we're looking around on the rim. Basically, we're on the southern side of the sinkhole and going to move around in a counterclockwise manner until our dive is ended.
The sand here is really smooth. I wonder how, it's probably pretty active, right? Like slowly rolling down the hill into the bottom of the sinkhole. I think eventually it would fill in. But maybe another cutthroat eel, dead center.
As you can see, we're looking down over the rim into the edge of this, from the edge of the sinkhole. Where the slope was relatively gentle compared to what we encountered down below. Looks to be a little shrimp in the water column there. Okay, that's good to know. It's just a vast expanse of flatness up here. Hey, take a look at that. Yeah, just a little pelagic animal. I guess it's yeah, it's. I think that was a Ketognath. Wow. Arrowworm floating by.
I think I just saw a colony just to the right there, but it'll be interesting to see if we're not going to see as many of the suspension feeders up on this flat area as we did down in that bowl, in that sinkhole. And if we don't, there must be some swirling currents down in that sinkhole to support those suspension feeders more so than there is up here on the flat part.
Yeah, I think that would be consistent with rippling we saw on the sand below, where here we see quite less. Ooh.
Yeah, I agree, Al.
There's a squid. Oh my goodness. We need to call the squid phone. Thank you. Let's take a look at this guy. Something else. Good. Is it? Oh, wow. Look how huge its eyes are. Come. No, we haven't got a name for the squid yet. I don't think, Mike. Hey, the squid is putting its.
Oh my gosh! Look how cool.
Yes.
You can see his Hi.
Mike?
Hey, yes, we do have a name for it. It's a Chiroteuthis.
Chiroteuthis. Awesome.
Yeah, it's a Chiroteuthis. I'm not certain on the species until I get a chance to look closely at the photophores. Well, we're getting close now. And then go back and look it up. The common one out here is Chiroteuthis varanii, but I'm not gonna go with that until I have a chance to go back and look at the literature.
Are these white dots around the eye? Those the photophores?
Those are all photophores around the eye. Yes, and it looks like
they're so perfectly symmetrical.
Well, it's developing an ovary in the posterior part. It looks like.
Is that the white you're talking about, or the brown?
I'm sorry?
The white you said was the ovary?
All the way at the posterior end towards the fins.
Okay.
I think that looks like ovary that's developing. And what you see, the ventral arms are extremely long, and they, like the mastigoteuthids, hold their tentacles in within a sheath in the ventral arms. because the tentacles are not very muscular. So they control the tentacles with their ventral arms.
You can see
Yeah,
it.
you can see it.
Oh my gosh.
And those are the ends of the tentacles that are sticking out beyond the tips of the ventral arms is what we're zoomed in on right now. It's not in its feeding position. It would extend those tentacles out much farther. And usually they drift along with the tentacles extended out, waiting for something to bump into them. And they have photophores on the tips of the tentacles, which probably function as lures.
So
That was
cool.
a nice observation.
Thank you so much.
Like it just drifted into the sinkhole.
My pleasure.
John said there was probably going to be a
I'll
lot of
hang up
squid.
until something else.
Yeah, no worries. Thanks for chiming
sorry?
in.
Yep.
What were you saying about the squid?
John was saying that he sent us an email this morning. He said a lot of the times around these sinkholes he sees lots of squid and therefore lots of swordfish. So I'm glad we got to see at least one.
Yeah, me too. All right, I'll talk to you later.
Thanks. Looks like another one ahead. Something ahead.
Bye.
At dead center. Another one maybe? Or is that a fish? That's a fish. Some type of eel or rat tail or very shiny. Oh, we got something red in the water.
Red in the water there? Almost. Oh.
He is very close. It looked like he had little legs running around.
So the whip coral that we had passed while we were... A couple people in the chat commented that it had small, tendrils coming off of it, and it reminded people of potentially of benthic ctenophores.
Oh, nice.
So might keep our eye open for that.
And we can look straight down this wall into the sinkhole.
Could we zoom in on that whip? Yeah.
Is this the same one?
I don't believe so.
Oh,
I think we've traveled too far, but still, might not be much to look at.
That first whip was when we were following the squid.
Okay.
We just saw the top curvature of it, so this would be a different one.
Of course, as soon as I go to get a shot of espresso, there's like awesome stuff to see. That happened to me once. I went to go get a cup of coffee, and there was a great white shark that I missed. I know. Everyone sends me out of the room.
That's nice.
Go. You can see this better. Is that just the coil at the end of a whip?
Yeah.
Branch there?
No, there's no branch. It's coming back around.
Okay, thanks.
Thanks for that. Look at it.
I was checking on that because this is what
Stick
a
up
very
these back
young
there.
trident candelabra would look like before it started to put out its branches. But I agree with you.
Yeah, we're good. Looks like a nice stick of pathies behind it.
Yep.
Did Mike put the name, the Chiropteuthis, into the...
I wrote it down.
Okay.
I put it in the chat, Stephanie.
Thank you.
Rims of, well, I've never been in a sinkhole before, but other rims of walls and so on are usually really good places for the suspension feeders because you get that, curved substrate acceleration over it. So it's nice to see those few right there.
For those of you who were with us when we saw the tripod fish, we were discussing whether they might be blind, but Ken Sulak says they're not blind, but, but have very, very small eyes. They basically sense their prey with a well-developed lateral line and the specialized pectoral fins held aloft. Really quite specialized way of feeding. But they do have the eyes, and those are probably used to detect bioluminescent prey.
Can you guys look at 6 o'clock? That might be some trash. 6 o'clock right off screen. There might be some trash right there, that black thing. Yeah, I think that's probably a piece of trash of some kind. Rubber, maybe?
Bummer.
Okay. Yeah, of course, it's a bummer. But...
Yes, we would like that last Niskin sample. I think it's perfect timing.
Yeah. So that's going to be sample number 10W.
Mm-hmm. This is for environmental DNA, basically loose DNA floating in the water, shed from the organisms that make their home there. Wonder if we'll get sargassum in this one, or maybe all of them.
Right?
Yeah. We're going to have to end our dive soon.
Yeah, sea state's picking up.
Deteriorating sea state. A little bit ahead of schedule, but we got quite a lot of great view of this sinkhole from top to bottom. Or I should say from bottom to top.
Yeah. Are we planning on having a post-dive planning call? I'm not sure. I thought the sea state was going to get so bad. I'm not sure we're going to even be able to dive. But no one has officially told me that yet. So.
Right.
Hey, it's Matt.
Hey, Matt.
Yeah, so we're going to transit tomorrow. The Lake Plateau is going to get so bad for the next few days. We're actually going to transit down towards the Keys and the West Florida escarpment over the next few days, and I'll possibly get 3 dives in that region. So that's the idea. And then we'll surf the Gulf Stream back up and try to get a few more dives in on the Blake later next week.
Awesome. So we're going to have at least a day, maybe 2 days without diving.
Correct.
Is that correct? Okay. So we'll have our pre-dive planning call in a few days for our next dive, and hopefully we all don't get violently ill for the next few days.
And if anybody on the call wants to submit suggestions for dive sites off of the Florida escarpment or Portales Terrace region, please feel free. We're going to take any and all suggestions right now and sort them out to see what we can operationally do in the next few days.
Awesome. Thanks, Matt. All right, so right before we come off bottom, and this is our last dive for a few days, we will keep everybody posted, though, when we get to go down again. Right now, we're 186 miles off the coast of Florida, along the Blake Plateau. We actually found this solitary blue hole that doesn't, or sinkhole that doesn't seem to be anything like this in the area at all. They're common near the coast, but we have not seen any other sinkholes in any of the multi-beam on the Blake Plateau, right? Mm-hmm. And then we started the dive at the base of this the base of this divot at 1,213 meters, and then we're going to end the dive at the top rim on the sedimentation around the outside of it at 1,140 meters. So that was a total of 67-meter rise. The walls at the base were very white and layered in parts, and then really dark and coated in ferromanganese crusts and others. But you could still see that really cool layering.
Those would be walls that were there standing for much longer time, probably, than the white exposed pieces.
Yeah, I would think, and you can even see the rocks that had fallen down. We collected 2 of those. Mm-hmm. Let's see. So then, as we came up, so at the base, at the bottom is mostly sand, and then as you come up the beginning of the wall at the bottom, it's very steep, 90, 80, 70 degree wall. And then as we come up, it gets a little more less steep until we get to the top, and that top rim is, you can see, just turns into sand and sediment. Looks like we're looking at now. So the highest slope actually in the multibeam was 52 degrees, but I would say it was when we actually physically look at it, it was probably steeper than that at the bottom.
Yeah, and we saw quite a lot of suspension feeders down in there, indicating that there was, and quite a bit more than we saw up on the rim. So, indicating that the water is moving and bringing nutrients to the to support those larger colonies.
Yeah,
suspension feeders.
And in the center of the sinkhole was actually pretty high current when we first arrived. But as you get closer to the wall, it gets the current drops off significantly, almost to nothing, it looked like, in parts where we could see the sediment not moving at all in the screen. We did have a few coral and a bunch of fish. We even saw a tripod, which was
really
they're
great.
so cool. They're just the neatest things. Anyway, we saw one of those, and that's the first one we've seen this cruise. We also saw that awesome squid, Chiropteuthis.
Yeah.
Anything else?
Chiropteuthis jubini is what Mike Vecchione came back and identified it as.
I will update my
And
notes.
one of the most exciting things was that we were able to collect this, an urchin group that has never been collected by NOAA Ocean Exploration via the Okeanos before. And so that is a really nice, exciting addition to the collections that are going to be housed back at the Smithsonian.
Anything else? Any other?
I'm going to throw out the Hilariid, which is that ribbon-like growing octocoral that has an external chitinous skeleton, and that's useful because when they're growing on rock, you can't pull off the bottom of the ocean, so they're hard to collect. So that was a really good find.
Nice.
Fantastic.
All right, good. So, we're coming up a little early today. Sorry about that. The sea state's picking up, and it's going to get really, really nasty out here the next few days. So I think we're going to make our way, Matt said, down to Keys and maybe the Portales Terrace. So if you have any input on our scientists ashore, have any place they want to dive off the Portales Terrace, please let us know. I know there's a few places we've been inside and outside the HAPC, but off the top of my head, I can't remember the depths. So they might be too shallow for a D2, because the minimum D2 can dive is actually 250 meters. Otherwise, it can overheat because the temperature in the water is too warm. Anything else?
I think that sounds good.
Okay. Thank you for diving with us today. You can follow our social media. So the Ocean Explorer, or NOAA's Ocean Explorer Twitter feed is at Ocean Explorer. They're also on Facebook at Ocean Exploration Research, and on Instagram at NOAA Ocean Exploration. And I'm on Twitter at Deep Sea Nerdy, and Facebook and Instagram at Deep Sea Nerd. And Alan is on Twitter at Collins_NOAASI, and on Facebook at Alan Collins Zoologist. So feel free to reach out to us if you have any questions. We're happy to answer them over Twitter. That's the fastest way to get to me. I don't really pay too much attention to my Facebook page. Sorry, guys. And I post pictures on Instagram sometimes. But so if you have questions or want to reach out to us personally, we're happy to answer questions.
Absolutely.
Just tweet us. No
Always
big deal.
Open.
We also, let's do a quick rundown. The way these. I am blanking today. I'm having my brain is just turned off. Okay. These sinkholes form is usually by the water seeping into the underlying rock. And that fresh water wears and wears and wears, and then eventually it just collapses. And this one's really odd because it's so far away from shore, so it must have happened a really long time ago. However, I feel like it should be filled in with more sediment. So I just I don't even know what to do with this place.
Right.
No idea.
Well,
so anyway, that's enough.
Hopefully,
I just ate espresso, drank espresso. So we're going to go process samples. Have a wonderful afternoon. Tweet us. And we will be back in a couple days. Check back in. If you follow Ocean Explorer on Twitter, I guarantee it'll let you know when we're diving again. So, have a good night, everybody. Hope you enjoyed. That was awesome.
Thank you.
That was an awesome dive.
It was a great dive.
Bye, guys.